Dear participants, welcome to this advanced course on the eukaryotic cell cycle, where we will explore the molecular, cytogenetic and epigenetic mechanisms that sustain life. We will critically examine how precise regulation ensures genome stability, how its dysregulation drives disease, and how modern experimental approaches and therapeutic innovations shape the future of biomedical research. Dear students, today's learning objectives are: we'll analyze the structure and phases of eukaryotic cell cycle, comprehend the molecular basis of cell cycle regulation and checkpoints. We will understand chromatin dynamics and nucleosome assembly and evaluate cytogenetic and molecular events of mitosis, meiosis, and fertilization. Later, we will critically assess cell cycle dysregulation in human disease, apply experimental techniques to study chromatin and cell cycle dynamics. Later, will integrate cell cycle knowledge with future therapeutic innovations. Dear students, the eukaryotic cell cycle is a highly regulated process that ensures faithful replication and segregation of genetic material, supporting growth, repair, reproduction, and genomic stability. From the discovery of mitosis under light microscopy to Howard and Pelk's identification of the S phase, research has revealed cyclins, cyclin-dependent kinases, and checkpoints as key regulators. The cycle comprises GAP1 for growth, S phase for DNA replication, GAP2 for error checking and mitosis for chromosome segregation, and cytokinases. Dysregulation such as mutation in p53 or retinoblastoma protein drives uncontrolled proliferation and cancer. Understanding these mechanisms underpins modern research, medicine, and therapeutic innovation. Dear students, moving on to the overview of eukaryotic cell cycle. The eukaryotic cell cycle is a regulated process ensuring accurate DNA replication, chromosome segregation, and genomic stability. Discoveries from Alma Howard and Stephen Pelk to model organisms like yeast and xenopus revealed conserved control by cyclins, kinases, and checkpoints. Mutations in p53 or retinoblastoma protein drive cancer, while meiosis and fertilization sustain heredity and life's continuity. Phases of the eukaryotic cell cycle is traditionally divided into 4 sequential phases with a quiescent state. Then G1 phase or GAP1. During this phase, the cell grows, synthesizes proteins, and prepares for DNA replication. Cyclin D and retinoblastoma protein are critical regulators, integrating signals from growth factors and stress responses to decide whether the cell commits to division. Then is the S phase, the synthesis phase of DNA replication. And DNA replication here produces identical sister chromatids for each chromosome. The replication machinery is supported by histone chaperones, ensuring proper nucleosome assembly behind the replication fork. Then is the G2 phase or GAP2. This phase allows completion of DNA replication, repair of any errors, and preparation for mitosis. Cyclin B and cell division cycle 2, that is CDC2, also known as CDK1, prepare the cell for mitotic entry. Then is the M phase, mitosis phase. Nuclear and cytoplasmic division produce 2 genetically identical daughter cells. Then we have G0 phase. Some cells exit the active cell cycle into a quiescent state, either temporarily or permanently, in response to differentiation signals or nutrient deprivation. Dear students, let's talk about molecular regulation of the cell cycle. The eukaryotic cell cycle is a meticulously orchestrated process governed by a dynamic network of molecular mechanisms ensuring precise DNA replication and division, which is critical for genomic stability and cellular function. Then if we see regulation of eukaryotic cell cycle is a sophisticated molecular network involving kinases, inhibitors and checkpoints that safeguard genomic integrity and coordinate division with growth. We have cyclins and cyclin-dependent kinases. Progression through the cell cycle is orchestrated by these cyclins. These proteins whose levels oscillate in a phase-specific manner, and cyclin-dependent kinases, serine threonine kinases that phosphorylate target proteins to drive phase transitions. We have cyclin D or CDK4/6. It regulates G1 progression by phosphorylating retinoblastoma, releasing E2F transcription factors to activate S-phase gene expression. Then we have cyclin E and CDK2 that promotes initiation of DNA replication. Cyclin A, CDK2-1, ensures completion of S-phase and entry into mitosis. We have cyclin B, CDK1, or maturation promoting factor, which controls G2 to M transition and triggers mitotic events. Dear students, we have cyclin-dependent kinase inhibitors, CK1s. CKIs such as P21, CIP1, cyclin-dependent kinase inhibitor 1, P27, KIP1, kinase inhibitor protein 1, and P16, INK4A, inhibitor of CDK4A, inhibits CDK activity to prevent premature progression. CKIs are activated in response to DNA damage or cellular stress, enforcing checkpoints that maintain genomic integrity. Moving on to cell cycle checkpoints. Checkpoints ensure fidelity. Checkpoints serve as surveillance mechanisms. Number 1, G1S checkpoint. It assesses DNA integrity before replication. We see activation of P53, the guardian of the genome, and can halt the cell cycle or induce apoptosis. Number 2, G2 to M checkpoint. It confirms complete and accurate DNA replication prior to mitosis. Then spindle assembly checkpoint, the third one, ensures all chromosomes are correctly attached to the mitotic spindle before anaphase, preventing aneuploidy. These surveillance mechanisms involving ataxia talgigacea mutated and ataxia talgigacea and RAD3 related maintain cellular homeostasis. Tumor suppressors and proto-oncogenes. Dear students, P53 detects DNA damage, activates repair pathways, induces apoptosis or halts the cell cycle. Mutations in P53 are found in over 50% of human cancers. Then we have retinoblastoma protein. It inhibits E2F transcription factors, controlling S-phase entry. Loss of retinoblastoma function leads to uncontrolled proliferation. Proto-oncogenes, genes that promote cell division. Gain of function mutation convert them into oncogenes, driving tumorogenesis. Dear students, moving on to chromatin dynamics and DNA replication. Nucleosome assembly. During DNA replication, chromatin must be reassembled into nucleosomes, the basic unit of chromatin. A nucleosome consists of 147 base pairs of DNA wrapped 1.65 turns around a histone octamer. H2A, H2B, H3, H4 with linker histone H1 stabilizing higher order structures. Histone chaperones like chromatin assembly factor 1 or CAF1 and anti-silencing function 1 ASF1 deposit newly synthesized histones behind the replication fork, ensuring proper nucleosome spacing. Students, let's talk about post-translational modifications of histones which regulate chromatin accessibility. Number 1 is acetylation. It neutralizes lysine charges, loosening the DNA histone interactions and promoting transcription. Number 2 is methylation. Site-specific effects. H3K4-ME3 promotes transcription, whereas H3K9-ME3 and H3K27-ME3 repress genes. Number 3 is phosphorylation. It regulates chromatin condensation during mitosis and repair. Last, ubiquitination, which modulates gene activation or repression depending on the context. These PTMs form the histone code, dictating chromatin function. Then moving on to DNA repair integration, proliferating cell nuclear antigen or PCNA, which coordinates replication and repair. While checkpoint kinases, ataxia telangiectasia and RAD3-related and attentia telangiectasia mutated monitor DNA integrity. DNA lesions recruit repair complexes to preserve genome stability. Dear students, let's discuss mitosis, the equational division. Mitosis ensures faithful segregation of sister chromatids. There are certain key events in mitosis. The mitotic phase precisely divides replicated chromosomes into 2 identical daughter cells, ensuring genomic stability through a series of tightly regulated stages. Number 1 is prophase. Chromosomes condense via condensin complexes and histone phosphorylation, while centrosomes separate through nucleate spindle microtubules. Number 2 is prometaphase. Nuclear envelope breakdown enables microtubules to capture kinetochores on centromeres with dynamic instability, facilitating attachment. Number 3 is metaphase. Chromosomes align at metaphase plate with kinetochore microtubules, generating tension. Then fourth, we have anaphase kinetochore. Microtubules shorten, pulling chromatids poleward, while polar microtubules elongate the spindle via kinesins. Then we have telophase. Chromosomes decondense and nuclear envelopes reform via lamin dephosphorylation and nucleoporin assembly. Nuclear pores restore, preparing nuclei for interphase. Then cytokinesis. The ectomyosin contractile ring in animals cleaves the cytoplasm, while plants form a cell plate via phragmoplast. This completes division, yielding 2 daughter cells. Then we see spindle and kinetochore function. Spindle microtubules from centrosomes connect to kinetochores. Proper attachment prevents aneuploidy, a cancer hallmark, ensuring accurate chromosome segregation. Dear students, let's discuss meiosis. Meiosis is specialized division reducing diploid cells to haploid gametes. Comprises 2 rounds, meiosis 1, the reductional division, and meiosis 2, the equational division, generating genetic diversity through recombination and independent assortment. Meiosis 1, the reductional division. Meiosis 1, the reductional division, halves the chromosome number from diploid to haploid, generating genetic diversity through homologous chromosome segregation and recombination. Meiosis 1 separates homologous chromosomes, reducing pleiadae from diploid 2n to haploid, that is N, distinguishing it from mitosis. This process, driven by cohesion and recombination, ensures genetic diversity through allele shuffling. We have prophase 1, leptothene stage. Leptothene initiates chromosome condensation, transforming diffuse chromatin into distinct threads visible under microscopy. Prophase 1, zygotene. Zygotene features homologous chromosome pairing via the synaptonemal complex, a proteinaceous structure aligning homologs. The synaptonemal complex protein 1. SYCP1 facilitates this pairing, which is critical for crossover initiation. Then prophase 1, spachotene. Spachotene enables crossing over at chiasmata, where SPO11 induced double-strand breaks are repaired by homologous recombination, yielding 50 crossovers per human meiosis. Then, prophase 1's diplothene. Diplothene reveals visible chiasmata as the synaptonemal complex dissolves, allowing homologs to partially separate while remaining linked. Then, prophase 1's dikinesis. Dikinesis fully condenses chromosomes with Aurora B kinase, enhancing compaction for spindle attachment. Then we have metaphase 1. Metaphase 1 aligns bivalence at the spindle equator with kinetochores of homologous chromosomes attaching to microtubules from the same pole. Then anaphase 1. Anaphase 1 separates homologous chromosomes via cohesion cleavage along chromosome arms, reducing pleiadae. Then telophase 1. Telophase 1 reforms nuclear envelopes around segregated homologs, often with incomplete cytokinesis in oocytes. Then meiosis 2, equational division. It mirrors mitosis by separating sister chromatids to produce 4 haploid cells, a critical process for generating genetic diversity in gametes. Meiosis 2, prophase 2. Prophase 2 condenses chromosomes into compact structures, preparing them for alignment, with centrosomes migrating to form the mitotic spindle. Meiosis 2 is metaphase 2. Metaphase 2 aligns chromosomes at spindle equator with kinetochores attaching to microtubules from opposite poles. Meiosis 2's anaphase 2. Anaphase 2 separates sister chromatids via cohesion cleavage at centromeres by separase, which is activated by anaphase promoting complex, cyclosome, APCC. Meiosis 2's telophase 2. Telophase 2 reforms nuclear envelopes around segregated chromatids, with decondensation facilitated by phosphatase activity. Then cytokinesis follows, yielding 4 haploid cells, each genetically distinct due to prior recombination and assortment. Dear students, moving on to fertilization and early embryonic development. We'll see certain key events in fertilization. Fertilization restores diploidy by merging these haploid gametes, initiating embryogenesis through recognition, fusion, and activation. Sperm preparation and penetration. In mammals, capacitation in the female tract enhances motility and prepare the acrosome. Binding to the zona pellucida triggers the acrosome reaction, releasing enzymes like acrosin for penetration. Then we see gamete fusion and activation. Fusion involves isomer-1 on sperm and Juno on egg, activating calcium oscillations via phospholipase zeta. This completes meiosis II, extrudes the second polar body and triggers the cortical reaction, preventing polyspermy. Then we see pronuclear formation. Sperm chromatin remodels, pronuclei fuse in syngamy, activating the zygotic genome. Then plant double fertilization. One sperm fuses with the egg to form a zygote, the other with the central cell to produce triploid endosperm. Dear students, let's talk about cell cycle dysregulation and diseases. Cell cycle dysregulation disrupts the precise orchestration of cellular division, leading to diseases like cancer and developmental disorders. For postgraduate students, understanding these disruptions reveals molecular mechanisms underlying pathology and therapeutic opportunities. If we talk about cancer, cell cycle regulation ensures precise DNA replication and segregation, but its breakdown underlies cancer development. Mutations in cyclin-dependent kinases, that is CDKs, cyclins or tumor suppressors like tumor protein p53 and retinoblastoma protein, abolish cell cycle checkpoints, drive uncontrolled cell proliferation, a hallmark of cancer. For example, overactive cyclin D, CDK4 to 6 complexes or TP53 mutations present in over 50% of cancers bypass the G1S checkpoint, promoting the unchecked DNA replication. Dysregulation of the spindle assembly checkpoint, SAC, mediated by proteins like mitotic arrest deficient 2, MAD2, leads to aneuploidy, fueling the tumor progression in cancers like colorectal carcinoma. Targeted therapies such as CDK4/6 inhibitors, for example, palbociclib, restore control in breast cancer, highlighting clinical relevance. Mutations in CDKs, cyclins or tumor suppressors result in uncontrolled proliferation. Dysregulation of checkpoints, especially SAG and G1S, contributes to tumor progression. Then aneuploidy and developmental disorders. Accurate chromosomal segregation is essential for development. Errors in mitosis or meiosis can cause aneuploidy, where cells harbor abnormal chromosome numbers, leading to developmental disorders like Down syndrome, the trisomy 21. Results from meiotic non-disjunction producing 3 copies of chromosome 21. It manifests with intellectual disability, distinct facial features, and increased risk of leukemia. We see Turner syndrome, monosomy of chromosome X. Affects female development. Females with a single X chromosome exhibit short stature, ovarian failure, and cardiac defects due to chromosomal loss. Then we have Rett syndrome. Unlike classical aneuploidies, Rett syndrome arises from mutations in methyl CpG binding protein 2, that is MECP2, disrupting chromatin regulation and neuronal function. It primarily affects girls, leading to developmental regression, motor dysfunction, and seizures. These examples underscore how cell cycle dysregulation links molecular events to disease phenotypes. Dear students, let's discuss some experimental approaches in cell cycle and chromosome study. Advanced experimental techniques are pivotal to unravel the molecular intricacies of chromatin dynamics and cell cycle regulation. These methods from mapping protein DNA interactions to visualizing real-time dynamics, drive discoveries in epigenetic and diseases. Number one, we have chromatin immunoprecipitation. It isolates DNA-bound proteins or modified histones using specific antibodies, followed by sequencing to map interactions. Assay for transposase accessible chromatin using sequencing or ATAC-seq. It identifies open chromatin regions by inserting sequencing adapters into accessible DNA via transposase. It highlights active promoters and enhancers, providing insights into regulatory landscapes during cell differentiation. Then we have chromosome conformation capture, HIC and 3C. It captures three-dimensional chromatin interactions by ligating spatially proximal DNA segments, revealing topologically associating domains or TADs. They elucidate enhancer-promoter loops, critical for understanding gene regulation, disruptions in disease like leukemia. Then we have live cell imaging and super-resolution microscopy. Dear students, such as stochastic optical reconstruction microscopy. It visualizes chromatin and cell cycle events like mitosis in real time with nanoscale resolution. These methods track kinetochore dynamics or nucleosome repositioning, enhancing studies of mitotic fidelity. Then we have flow cytometry. It measures DNA content using fluorescent dyes like propylium iodide to determine cell cycle phase distribution, like G1S, G2 to M. We'll see future directions and therapeutic applications. Chromatin-targeted therapies. Histone deacetylase inhibitors, HDACs, and DNA methyltransferase inhibitors are used in cancer therapy. CRISPR-based chromatin engineering enables manipulation of gene expression and epigenetic states. Systems biology approaches integrate genomic, epigenomic, and proteomic data to model cell cycle regulation comprehensively. Dear students, as we conclude our exploration of eukaryotic cell cycle, the mitosis, meiosis, and fertilization, remember that mastery of these core principles is not only essential for examinations, but also forms the intellectual foundation for advanced research and clinical innovation. I encourage you to keep questioning, connecting molecular details with broader biological phenomena, and applying this knowledge as future scientists and professionals shaping tomorrow's discoveries. Thank you, dear students.